a video of it, just so you get somebody else besides me explaining it. And it also hopefully kind of has our carbon chasing game going on as well.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during the oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. Initial steps in glycolysis are the additions of two phosphates to the glucose molecule at the expense of two molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and two low-energy adenosine diphosphate molecules, or ATP. This 6-carbon sugar diphosphate molecule is then split into two 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme Pe+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
So what does he mean by aerobic or anaerobic conditions? What does that mean? Yeah, aerobic and anaerobic in science-ese means with or without oxygen. So we are very much right now following the aerobic pathway. We're going to talk about glycolysis and then move into the other things you do if you're using oxygen. Okay? But what he's mentioning is that since everybody does glycolysis, the next step isn't going to be the same for those who don't use oxygen. Everybody does glycolysis, but this is a branch point where if you're not an oxygen user, you're going to branch into something else after glycolysis. We'll talk about that after we're done with the aerobic pathway. That'll be in the next couple of days. Any questions about glycolysis before we move on? Okay, well, here comes then the Krebs cycle. I think we're going to start with the same kind of video, but this time about the Krebs cycle. This one, I think. Kind of the carbon chasing version of the Krebs cycle. And I'm not going to make you draw it like I did with light independent reactions. Let's just kind of get a sense of where we are.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon the acetyl CoA enters the Krebs cycle, which occurs in the mitochondria. During the conversion of pyruvate to acetyl CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form ADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule, initially reacts with acetyl CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, I know it looks like a lot, but understand you don't have to memorize this diagram. here are the high points of what's going on here. First of all, just in terms of carbons, notice that what's happening here is pretty simple. We're taking a molecule that was made from like pyruvate and this 4-carbon molecule right here. And we are just gradually shaving off CO2s, right? CO2 comes out here, CO2 comes out here, CO2 comes out here. At that point, you've basically broken down the pyruvate. If you think of the pyruvate as a 3-carbon molecule, boom, boom, oops, sorry, boom, 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 and we're done. But it's in the rearrangement of all these weird little carbon molecules that we load up the babysitters with electrons. And I know the worst part is, oh my God, one more babysitter? Yes, one more babysitter. It's called FADH2. It's not any different for our purposes than NADH or NADPH from photosynthesis. It's just an electron carrier. The point of this whole thing here is to take pyruvate and use its high energy bonds to load up electrons onto electron carriers. Yeah, you get a couple ATPs, just like you did in glycolysis, but they are small potatoes compared to what's about to happen with all these babysitters that we've made. Questions? We're going to write this down. We'll write down what you need to know here, but while you're looking at it, is there anything that totally doesn't make sense? You know, less detail here than light independent, like you don't have to know 3 phases or anything like that. What you need to know is pyruvate's going in, CO2 is getting shaved off, and we're making a couple ATP and a bunch of electron carriers. Krebs cycle, by the way, is sometimes called the citric acid cycle. You don't have to necessarily memorize that. On the off chance you see an old textbook, they might call it that. The citric acid is one of the little 4-carbon molecules involved in it. In the Krebs cycle, physically what's going on is the pyruvic acid is broken down into CO2s. You get a bunch of NADH and FADH2, which are high energy electron carriers headed for the electron transport chain. You get 2 ATP. Reactants and products. Pyruvic acid goes in, and the low energy forms of our 3 high energy molecules, FAD, which is how they write it. If it makes more sense to you to say FAD plus, I don't care. I'm kind of wrong. NAD plus and ADP plus P.
When pyruvic acids are broken down into CO2, what is the
Which hydrogen? Oh, it's still there. Well, okay, good question. Some of it might be given off as hydrogen ions. I don't know. Good question. Not important, but good thinking.
Well, there's C3 and O3, but if it's being broken down into CO2 molecules, then what are the...
That's what she was just saying. And it may be that it stays part of the thing it's getting shaved off of. And I don't know the full reaction diagram there, so I can't tell you. I'm sorry. The biochemist could, I can't. Products, we get, of course, CO2 coming out, but then the 3 high energy molecules. And now we are ready for the real energy moneymaker of the cell.
Why is there only one NADH and this one FADH?
Different molecule. It's chemically quite closely related, but they are different. And so, one's carrying more hydrogen than the other. And so these two babysitters, NADH and FADH2, now take the spotlight, and they are going to head to the inner mitochondrial membrane, which is where the electron transport chain is. And here's where things start to look really familiar, if you've remembered the details of the light-dependent reactions. Because what's going to happen is the NADH and FADH2s are going to float over to, and okay, so here's a mitochondria. Mitochondrion is the singular. And remember, it has two membranes. Well, we can think of the mitochondria as basically having two spaces. It has the matrix, which is the space in the middle, and then it has the intermembrane space, which is the space in between the two membranes. And so this, which is the electron transport chain, is actually kind of embedded in the inner mitochondrial membrane. And for us, we're going to say this is the intermembrane space. That's this space here. And this is the matrix, if you want to think kind of inside and outside. So in comes NADH and drops off its high energy electron and becomes NAD+. And now we have an electron here. Does this look familiar? Then the electron bounces, going, and winds up at the end. And what happens at each step? What goes where? You remember what happened in photosynthesis? Protons get moved. And so protons in this case are going to be being moved from the matrix into the intermembrane space. Protons get moved from the matrix into the intermembrane space. And just like in light-dependent reactions, they build up and build up and build up until there's a whole bunch of them. And just like in the light-dependent reactions, there's a machine over here called the ATP synthase. Yeah, familiar? So enough of those electrons bounce through this chain to build up enough of these protons, and we've got enough pressure so that these protons will start to come out this way. When they come out this way, that machine, remember, is built so that it takes ADP and a phosphate, smushes them together, and makes ATP, which is the whole point of this, right? We want ATP, and you get a buttload of ATP from the electron transport chain. So, if you think of the electron carriers as the babysitters, this is the treadmill that powers your house for a week when you drop your little hyperactive brother on it. And it really does make a ton of ATP. These electrons jump along those proteins stepwise, moving protons in the intermembrane space the whole time. So protons get pushed into the intermembrane space. The ATP synthase is the water wheel that uses the force of those protons escaping to build ATP. I keep saying a buttload. Well, it turns out a buttload is roughly 32 ATP per glucose. Keep in mind that we got 2 from glycolysis, 2 ATP from the Krebs cycle. Now we're getting 32 out of the electron transport chain. So clearly, this is your big energy producer. Okay, here comes the fun part. Fun for me, really. Fun for me because I love when I get to teach you something that you didn't know. Questions you never realized that had deeper answers. So here's the problem. The problem is that we don't have, like, remember in photosynthesis, in the light-dependent reactions, we had the electron jumping and then getting exhausted. but then it wound up in a new chlorophyll complex and got re-excited, right? We don't have that here. We have an electron that's jumped down the chain and is now absolutely exhausted and stuck. And if it remains stuck there at the end of the chain, the next electron to come down the pipe is going to get stuck here. And then the next electron is going to get backed up and stuck here, and so on. And we basically have a traffic jam of electrons. Something has to come in and drag this exhausted electron off of this protein that actually has quite a bit of attraction for it. Like it sticks to it pretty well. So we need something especially electronegative, something that's very good at attracting electrons. And this was a huge step in the evolution of cells to be able to evolve to use this particular gas instead of being killed by it. It's a very reactive gas. In fact, it oxidizes just about everything. And so to be able to use this to pull the electrons off was a huge step. This is one of those things you know you need. You just didn't know why up until now. What is the thing that pulls those electrons off the end of the chain? Anyone? This is it. This is where oxygen, that's O2, which is a gas, right? Oxygen comes in. It's highly electronegative. It's very good at grabbing those electrons. And in fact, one O2 molecule will end up grabbing 4. All right? Time to go? Oh, 2 weeks. Okay. Need to walk? Sailor, you want to go?
Sure.
Thank you. One O2's worth of oxygen will grab 4 electrons and actually grab 4 protons along for the ride. What's 4 electrons plus 4 protons? What's that going to make, Nevada? Yeah, 4 hydrogens. What's 4 hydrogens plus 2 oxygens? It's going to be 2 water molecules. Yeah, so oxygen is your final electron acceptor. I don't want to oversell this, but you've spent 15 years of your life not knowing why you have to breathe. It's kind of like the question, what does DNA do, right? You were like, it's what makes you you. But what does it do? We'll get deeper in that. But now you can say, like, okay, why do we need oxygen? Well, we need it to live. Why? Because, Mom, it's the final electron acceptor for the electron transport chain. If we didn't have oxygen, it would back up like a traffic jam and get filled up with pouty, exhausted electrons and shut down. Oxygen is what keeps your electron transport chain going.
So that's why people do grab. It spends some weeks like around like they quickly.
Yeah, you're not getting that. Suddenly, you're not getting the 32 ATP anymore. You're only getting the 2 and 2 from glycolysis and Krebs. Now we'll talk about, because you do run out of oxygen a lot when you exercise, and you do have an alternative pathway that you switch to that's fast enough to keep up for a while. Think sprinting, not very long, right? We'll get into it. But yeah, this is why you got to breathe. You got 2 whole systems of your body, circulatory and respiratory, just to deliver enough oxygen to every cell in your body to keep your electron transport chains going. So again, reactants and products, NADH and FADH2 are electron carriers. Oxygen, our electron receiver. And of course, a lot of ADP plus P to make those 32 ATPs. Thank you, Sam. Products, tons of ATP. Water, which makes more sense this time because it is the product of the oxygen grabbing the electrons and protons, and then the used up versions of the electron carriers. If you're to this point feeling like you have a pretty good handle on everything we talked about, good job. If not, you're normal. This is not easy. Okay, but it is doable. I promise you can do this. We're not expecting anything that you're incapable of. And we're not going especially deep in most of these things. Questions at this point?
Go back to the previous slide for a second.
In a minute. Everybody else caught up? Any other questions? So yeah, we did glycolysis and the Krebs cycle to generate ATP, but only kind of. We got a little bit of ATP out of them. What they really do is set up electron carriers to deliver electrons to the electron transport chain.
Where's E?
Can you clarify that?
The letter E for the.
Oh, did I skip it?
Is that just not a letter on there? Okay.
Yeah, I just skipped it because that's how my alphabet goes. A, B, C, D, F.
I'm not judging. I was just
Q.
curious for a second.
They were judging.
What is it?
Any other questions? I'm going to give you a pre-made flashcard. You know, like I had you fill in one side on the other flashcard. This one side's already done. The other side's intentionally blank. Don't fill it in. Use it as a flashcard. Use, you know, the blank side to try and figure out what goes in the blanks, and then check yourself with this. But that's it. I'm not going to put any more work on.